This is the second session of the MS and COVID-19 special Zoom conferences on of this August and September. We hope you're well and safe. I'm actually talking to you from lockdown Sydney in Australia at the moment. So, we're not all safe yet. We have a great panel of speakers today, and we're going to start right away because the program is full. As our first speaker is Celine Louapre from France. Thank you, Celine, to join us. And Celine and Michella are going to talk about serological responses after COVID-19 vaccines in MS patients. Thank you.
Hello, everyone. So thank you for inviting us to present the results of our serology study. So we're going to be two to present this study, myself, Celine, and Michella Ibrahim, who is a fellow at Paris Brain Institute. So, the study was recently published in GNNP, and so we investigated the serological response after SARS-CoV-2 infection in patients with multiple sclerosis or neuromyelitis optica spectrum disorders. And this is a study which was conducted across all Parisian MS centers, so which are listed here on the slide. So just speak of a bit of a background. So we published a few months ago, almost a year ago now, that from the COVISEP registry, which is the French registry of patients with MS and COVID-19, that neurological disability, age and obesity were risk factors for COVID-19 severity, and most critically ill patients were not receiving any DMT in our cohort. And then the Italian MS registry and the iWiMS registry from North America released also data that showed that DMTs targeting B lymphocytes and corticosteroids were associated with an increased risk of severe COVID-19. So then we wondered beyond COVID-19 infection, how patients were reacting and how the immune system was reacting, especially in patients receiving immunosuppressive therapy. And we already had from case series information that patients, especially on anti-CD20, were likely to have a lower seroconversion rate compared to patients on other DMTs. So we decided to conduct a prospective study to evaluate the seroprevalence and the level of several antibodies against SARS-CoV-2, anti-S for anti-spike and anti-N for anti-nucleotide peptide immunoglobulin in a cohort of patients receiving or not a DMT. So this is a monocentric study in the sense that that all the analyses were performed in a centralized laboratory at PT Salpêtrière Hospital, and all patients from Parisian area were involved and proposed to participate to the study. We identified the patients from the COVISEP registry, and then we collected blood sampling. It was collected as soon as possible, but at least 21 days after the onset of infection. But for some patients, the blood sampling, as this study was started only in October, some patients had the infection a few months earlier. And then we also plan, and this is something ongoing, to collect blood sampling at 6 months and 12 months follow-up. So the inclusion criteria were patients who had a COVID-19 infection, obviously, either biologically confirmed or with very typical symptoms, or CT scan abnormalities. And then we quantified the the levels of antibodies, so with a quantitative analysis for anti-S and semi-quantitative analysis for anti-N and anti-S IgA antibodies. And we also wanted to assess the level of IgG at total IgG, IgA, and IgM, just to have a reference, especially if some patient had hypogammaglobulinemia. So this is just the study population, but Michella is going to detail you all on that. So we identified 334 patients living in the COVID area from the COVID registry. So we asked them if they were willing to participate in the study and to have a blood sampling, and 119 patients decided to participate in the study. 115 patients with MS and 4 patients with NMOSD. Patients were diagnosed between February 2020 and February 2021, but blood samples were collected between November and April 2021. So I leave it next to Michella.
Thank you, Celine. I'm gonna share my screen. Or
I can go with the slide if you prefer, as you like.
As you wish, okay. So I'm going to present the results of our study. In fact, overall, all of our patients met the criteria of COVID-19 infection. When APCR was performed, it was positive in approximately 95% of the patients, and it was negative in 5% of the patients. However, in this patient, they had a positive serological testing that was performed as a standard of care by their treating neurologist. And all patients who did not have APCR had a typical COVID-19 presentation with typical symptoms, mainly anosmia. Next slide, please. As for the demographic and the disease characteristics of our population, we analyzed overall 115 MS and 4 NMOSD patients. They had a mean age of 43 years. For the MS patients, they had mostly a relapsing remitting form of the disease and a median EDSS of 2. They were treated with various DMTs. 21 had no DMT at all. 21 patients had an anti-CD20 therapy, whether it was ocrelizumab, rituximab, or ofatumumab. And 77 patients had various other DMTs other than anti-CD20. As for the COVID-19 infection, the symptoms were classical symptoms. They were the common symptoms that we find in the general population. Most common symptoms being asthenia, anosmia, and fever. The majority of our patients had a mild form of the illness. They required no hospitalization for 95% of our patients. The patients that were hospitalized were a total of 7. And only 3 of them required supplemental oxygen. Next slide, please. And these patients who required hospitalization for COVID-19 received the following DMTs. 2 were receiving ocrelizumab, 1 was on rituximab, 2 on dimethyl fumarate, 1 on teriflunomide, and 1 had no DMT. As for the serological results, we did a blood sampling after a mean of 5 months after the infection. And overall, approximately 80% of our patients had at least 1 positive SARS-CoV-2 immunoglobulin serological testing. And when we compared the different DMT groups, we found no difference in this rate. However, only the anti-CD20 group had a lower seropositivity of immunoglobulins and a lower titer. This is why, next slide please, we further investigated this, and we divided our population into 3 groups. We had 1 group who had no DMT at all, 1 group who had a DMT other than anti-CD20, and 1 group who had an anti-CD20 therapy. And here we can see in this figure that 95.2% of patients who had no DMTs had at least 1 positive immunoglobulin serology. 85.7% of patients who had a DMT other than anti-CD20 had a positive serology, and only 47.6% of patients on anti-CD20 had at least one positive serology. And this decreased rate was in fact comparable for all subtypes of immunoglobulin that were tested, whether it was anti-N or anti-S. Next slide, please. And this was further confirmed when we looked at the quantitative studies, when we looked at the titers of anti-S immunoglobulin G. We looked at the different subgroups that we analyzed, and here we can see at in this figure the different subgroups, and in the dashed line the threshold for a positive serology. And we can see that the mean of anti-S immunoglobulin G titer was only 1.4 in the anti-CD20 group. However, it was 2.4 in the group of patients who received the DMTs other than anti-CD20, and 2.7 in the group of patients who had no DMTs. We also did a linear regression analysis to see the association between the time from COVID-19 and the anti-S immunoglobulin G titer. And we can see clearly in this figure that the anti-CD20 group had an increased rate of decrease of the titer over time. So we see that the titer of anti-S immunoglobulin G was decreased only in the group who received anti-CD20 therapy. We also did a multivariate regression model where we looked at different independent variables and their impact on the likelihood of having a positive serology. We looked specifically at the groups of DMTs, the total immunoglobulin G, the time from COVID-19, the sex, the age, the hospitalization for COVID-19, and the obesity. And in this analysis, only the anti-CD20 had an impact on the odds of having a positive serology. It was a negative impact with an odds ratio of 0.07. We did this exact same analysis with the same independent variables to see the association with the titers of anti-S immunoglobulin G. And here we found a negative association with anti-CD20 and with hospitalization for COVID-19, and a positive association with the total immunoglobulin G. We also see a positive association with obesity, but this, however, was not significant. Next slide, please. And lastly, when we look at the time between the last anti-CD20 infusion and the COVID-19 infection, we see that this time was significantly longer in patients who had a positive serology. We see here that the mean was at 3.7 months in the seropositive group, compared to a mean of 1.9 months in the seronegative group. And this finding is actually interesting because it can be helpful when guiding the decision of the timing of the vaccination in this population. In conclusion, our study shows overall that people and patients with MS or NMOSD who were treated with anti-CD20 therapy had a significantly weaker serological response. And we don't know the long-term clinical consequences of this. This is why they should be carefully monitored in the future. Thank you for your attention, and we are ready to hear your questions.
Thank you, Celine and Michella. Do we have any questions? I'm looking at the chat. Yeah, well, I actually have one question. You know, I was quite interested by this negative association between hospitalization and spike IgG titers. And of course, you had one CD20 in there. But did you look in more particular in the other patients that were hospitalized? And did those patients on fingolimod or the other one in your groups had very, very low titer?
I think we should be very careful about this variable hospitalization because it's very, very few patients and some of them were on anti-CD20. So even using multivariate models, it's not really removing the effect of the interplay between the two variables. So I would not really go deeper into that, but I think in the general population, I think that the association was the opposite. Or
yes, it is.
Yes, so
yeah, that's why I was interested.
No, I think it does. Yeah, I'm not sure that we discussed that in the paper, but that's why I was thinking when I was wrapping up those results, and I was thinking, oh, okay, it's different from what is expected from the literature, but it's also completely driven by our 3 or 4 MS patients on anti-CD20.
Yeah, thank you.
I had a quick question, although this is so not my area of expertise, but I'm just curious, and I'm obviously following the wider literature. Any thoughts on, or do you have the capability of looking at any of the T-cell responses, or maybe Amit's going to talk on this in a moment, or what are your observations from the wider literature as to what you might find?
So unfortunately, we don't have cells. We just have serum samples because patients were having their blood drawing at home by a nurse, and it was much easier to just collect a blood sampling and just extract the serum. At that moment, there was really a lot of COVID cases. So people did not really want to go to the hospital, but I completely agree that I think that's going to be data that will be available from vaccine studies. Some centers, including are really collecting data on T-cell response. So,
yeah, thank you. Yeah, there was a question in the chat saying, do we have vaccine info for those specific patients? Abi and Shira, is that what you meant?
In any case, people in that study who are presented here did not have vaccine yet, but some of them will have vaccination before their second blood drawing or before the third blood drawing. So we will have data about what's happening on people with previous COVID infection and then who got a vaccine injection. But at that time in April, 1st April, none of the patients had been vaccinated. Actually, they could not be included in the study if they had vaccine in between.
Okay, thank you. So thank you, Celine. Thank you, Michella. It's very great.
You're welcome.
So I will go directly to our next speaker, Joe. I would like first, before you start speaking, I would like to remind everyone that if you are to use any part of this talk or discuss results that were presented here, I would like iWiMS recommends that you acknowledge the speakers and the source of the data. So thank you, Joe. The next one.
Thank you very much, and thank you to the iWiMS organizers for the invitation to be able to present our study. This is a study that was led by Riley Beauvais and myself, along with our colleagues listed here, where we've been in the process of completing a comprehensive analysis of both antibody and T-cell responses to the SARS-CoV-2 vaccines in patients on a variety of different disease-modifying therapies. For the sake of time, I didn't include background information, but the overall rationale of this is that we know that both the humoral as well as cell-mediated arms of immunity are important for protection from COVID-19. And we also know that the different types of DMTs that we use in MS treatment can differentially affect these two arms of immunity. And so we wanted to perform this comprehensive analysis. And we recruited a cohort of 80 subjects comprised of healthy controls as well as clinically definite MS patients who were aged 18 to 75. None of the patients or subjects included had a known history of known COVID-19, and all were unvaccinated at the time of enrollment. The study design included a baseline blood sample collection prior to vaccination, as well as blood sample collection 2 weeks following the second mRNA-based vaccine, or in the case of the adenoviral Johnson & Johnson vaccine, 4 weeks after the single vaccine. The immunologic outcomes of our study included antibody analysis, which was semi-quantitatively analyzed using a Luminex bead-based assay to the total spike protein, as well as the spike receptor binding domain, IgG. In addition, we used a phage-based display using VirScan technology to assess the antibody binding to different determinants within the spike protein. We also completed T-cell analysis where we stimulated PBMCs with pools of spike peptides, which allowed us to look at CD4 as well as CD8 T-cell activation using activation-induced marker expression, as well as intracellular cytokine secretion. We also completed tetramer analysis to previously published spike determinants. And this allowed us to perform ex vivo quantification and phenotyping of CD8 T cells following vaccination. This is the overall breakdown of our cohort. You can see the numbers of subjects in each cohort vary somewhat, as low as 5 to as high as 22 in the ocrelizumab group. The untreated and glatiramer treated patients were a little bit older than the other cohorts, which is not surprising given our treatment practices typically include patients who are older in these categories. The majority of patients in all cohorts were female, and this shows the breakdown of vaccine type based on Pfizer, Moderna, and Johnson & Johnson. Only 2 out of the entire cohort received the Johnson & Johnson vaccine. The blood samples were collected prior to vaccination, on average about a week before the first vaccine, and then on average 14 or so days following the second or final vaccination. This shows the antibody levels, both pre as well as post-vaccination. And then the top graph here shows the total spike antibody responses, and the bottom, the spike RBD. And what I think you can appreciate here is that the healthy controls, untreated MS patients, and then those treated with glatiramer, DMF, and natalizumab all showed significant increases in total spike as well as spike RBD antibody levels following vaccination. In addition, we found that those patients who were on S1P receptor modulators, rituximab or ocrelizumab, showed variable degrees of antibody production, which on average were significantly reduced compared to untreated patients. It is worth noting, though, that while we see some individuals with little to no antibody response, some of these individuals have quite high and near normal antibody responses. In looking at seropositivity based on the presence or absence of antibody against the total spike or spike RBD region, one can see that all of the groups indicated here, healthy controls, untreated MS, glatiramer, DMF, and natalizumab treated patients, were 100% seroreactive to both the total spike and the spike RBD. In contrast, we see varying degrees of seropositivity in the S1P and anti-CD20 groups. Interestingly, the rituximab group was the only group where we saw a significant reduction in total spike IgG seropositivity compared to untreated patients, with more moderate degrees of seropositivity in ocrelizumab. And I'll comment in just a few minutes of why we think this pronounced difference in the rituximab group was observed. A number of groups have already noted and reported that the time interval from the last anti-CD20 infusion prior to first vaccination has been correlated with vaccine-induced antibody levels. And so we wanted to also look at this in our cohort. And what we're showing here are on the x-axis are the months from the last prior anti-CD20 infusion before first vaccination, and the levels of total spike and spike RBD. And while there perhaps is some degree of a relationship here, in our cohort, we did not see a significant correlation. And so we looked at other variables that could be related to these antibody levels within the anti-CD20 treated patients. Shown here are the percent of CD9 positive B cells at the post-vaccination time point, and breaking this down between seronegative and seropositive individuals for both total spike and spike RBD. And you can see that those who were seropositive had on average a significantly higher level of CD19 positive B cells at this post-vaccination time point compared to their seronegative counterparts. We also looked at the cumulative duration, that is the total time patients have been on anti-CD20 therapy prior to their vaccination. And again, broke this down by seronegative and seropositive patients for both the total spike and spike RBD antibodies. And one can see that those patients who were seronegative to these antibodies were on average on anti-CD20 therapy significantly longer than those who were seropositive. And when we compare the rituximab to ocrelizumab cohorts, we observed that the rituximab treated patients have been on average significantly longer compared to the ocrelizumab treated patients. And so we think this likely explains why we see reduced seropositivity in rituximab treated patients, simply because these patients have been on anti-CD20 therapy longer. And in some instances, these patients have been on therapy for 10 years. We also wanted to perform a qualitative analysis of binding of spike-specific antibodies using the VIR-Scan technology. This platform involves taking peptides that span viral proteomes, and which can be basically engineered into bacteriophages that express these peptides, which can then be probed against serum. And this serum is then used to immunoprecipitate phage that it binds, and then those phage can be sequenced to determine which portions of the protein are the antibodies are reacting against. And so what I'm showing here are, this is for the spike protein only. This is basically spike protein for SARS-1 and 2. And shown here at the bottom are the regions of the spike protein, the N-terminal domain shown here at the left, the receptor binding domain here shown in red, and then towards the C-terminal region. Just one thing to note, the receptor binding domain is not well represented by this kind of technology. We think this has to do with the fact that the peptides used here are linear peptide sequences, and that the antigens formed by the RBD are probably three-dimensional and conformation dependent. And so we don't see those kinds of antibody responses as well with this technology. The left-hand side here shows the proportion of individuals who are show a detectable signal at these different regions of the spike protein. And this is broken down by the individual cohorts. The right-hand plot is the signal intensity, a heat map of those, where each row shows an individual patient. And I think you can appreciate a few things here. One is that anyone who is seropositive, we can see that essentially everyone has seroactivity along the C-terminal region, as well as regions flanking the receptor binding domain. But I think one thing that was really interesting that popped out to us is that even in individuals who were seropositive in the anti-CD20 and S1P receptor cohorts, we don't see the same breadth of reactivity across these different immunodeterminants. And so what we think this suggests is that even those who do mount a positive antibody response, including to the receptor binding domain, don't necessarily develop the same degree of a breadth of reactivity across different regions of the spike protein. And what this means from a clinical perspective and the biologic underpinnings for these changes are not entirely clear. We also looked at CD4 and CD8 T cell responses, as I said earlier, using both activation-induced markers as well as cytokine expression. Shown here is just a representative analysis of how we can identify CD4 T cells. This is from pre-vaccination in the top row, and from the same individual post-vaccination. We use CD137 and OX40 for the AIM analysis of CD4 T cells, as well as CD137 by 69 for CD8 T cells. And one can see that across all groups, for both CD4 and CD8 T cells, we see that we can readily identify spike-specific T cells following vaccination. In some of these instances, the post-vaccination analysis was not significantly higher than the pre-vaccination, but in all instances, there were similar levels of post-vaccination CD4 and CD8 T cells between all treated MS groups and untreated MS. One to note, though, is the S1P CD4 response here was lower post-vaccination, and this is consistent with the known CD4 T cell lymphopenia that occurs with S1P receptor modulators. We also looked at cytokine analysis shown here. This is just the post-vaccination analysis I'm showing to the indicated cytokines here. Most vaccination studies have indicated that interferon gamma, TNF, and IL-2 are the predominant cytokines produced by spike antigen reactive T cells, and one can see that the array, the polyfunctionality of these spike reactive CD4s as well as CD8 T cells is quite similar across all groups. And if anything, we actually saw a trend towards increased frequencies of cytokine producing cells within B cell depleted patients compared to others. And actually, in the case of interferon gamma producing CD8 T cells, this was significantly higher in rituximab treated patients compared to their untreated MS counterparts. And finally, this is just showing our tetramer analysis. This was done on about half of our cohort, where we attempted to basically match by concordance of HLA. And one can see here, this is all ex vivo, where we can enrich and purify these tetramer positive cells straight from PBMCs. And actually look at their memory status. This is just showing the tetramer positive cells are overwhelmingly effector memory in this case, compared to the tetramer negative cells, which include both naive as well as TEMRAs and a small portion of central memory cells. The bottom left-hand graph here is showing the proportion of tetramer positive samples that were detected from each cohort. And so this ranged from anywhere from about 25 to a little more than 50% of CD8s were tetramer positive across all the samples tested. And when we looked at the magnitude of these responses, although these are somewhat variable, and a lot of this has to do with immunodominance based on the HLA restriction, one can see that for the most part, the frequencies of these spike-specific CD8 T cells were essentially the same across all treatment cohorts. And finally, we looked at the memory status of these tetramer positive cells, and we can see that as one would expect, the overwhelming majority of these spike-specific CD8s are effector memory, with a smaller fraction representing central memory and TEMRAs. So just to summarize what we've shown that in untreated MS patients, as well as those treated with glatiramer, dimethyl fumarate, and natalizumab, had quantitatively normal total spike and receptor binding domain specific IgG responses. However, these spike antibody levels were reduced in patients treated with S1P receptor modulators and anti-CD20 monoclonal antibodies. And we showed in anti-CD20 treated patients that seronegative status was associated with reduced B cell counts at the time of post-vaccination analysis, as well as a longer cumulative treatment duration. We also found that in those who S1P and anti-CD20 patients who were seropositive, in VIR-scan, we observed that there was a restricted binding against certain immunodeterminants across the spike protein. And finally, we showed that spike-specific CD4 and CD8 T cell responses were overall quite similar across all MS groups, looking at both activation markers as well as expression of cytokines and memory status. So again, thank you all very much for your time, and I'm certainly happy to take any questions.
Joe, this is Amit. Can I ask a quick question?
Please. Yes, of course.
Great data. Thank you very much. Has to do with the S1P receptor modulator spread of the data, even with that early antibody slide.
Yeah.
You gave some rationale for why one might expect that with anti-CD20, not just the difference between Rituxan and ocrelizumab in terms of duration, but perhaps also timing from the last infusion. But with a daily introduced therapy like S1P receptor modulation, is it a time on drug related heterogeneity, or do you have a sense?
We did look at time on drug. I didn't show it here, but we did do it. And so the issue is there's only 7 patients in our S1P cohort. And so I'm a little leery of drawing too strong of a conclusion. That being said, the 2 patients, S1P treated patients who had the highest spike antibody titers were on it the shortest. So I do think treatment duration could be related, although I don't think we can definitively say that. But the other thing is I think this highlighted to me here, and I'm sure this is not a surprise to you, is the importance of T cells in generating a germinal center response. And even though these T cells are being retained in lymph nodes, they're not adequately getting to the germinal center, and so that's why I think we're seeing these reduced antibody titers.
Thanks.
Thank you, Joe. There's a question in the chat. Any data on cladribine treated patients?
Not from us. We have so few patients on that therapy, and so we focused on patients who on the more commonly used therapies in our clinic.
Thank you. I don't see any more question on the chat. So maybe, after Amit, maybe we'll have a bit more discussions between all the speakers. So thank you, Joe. So our next speaker is Amit Bar-Or.
Thank you. Thank you, Fabienne. Can you see the slide?
Yes,
Perfect.
I can.
Great. So thank you for the kind invite, and my job is made much easier after those 2 excellent presentations. And some of what I'll do is try to highlight where the data that you'll see here may complement, perhaps extend some of the previous, as well as, of course, reinforce some of it. The disclosures are shown here. And so I'll actually try to speak to 2 themes. One, of course, is along the same lines of testing vaccine competence and looking here also at both humoral and cellular responses, focusing on the mRNA vaccines in patients with MS on anti-CD20 therapy compared to healthy controls. But the other is this prospect of what's been referred to as analytic vaccination, where as we perturb a heterogeneous population's immune system with the same intervention, it could be a treatment, in this case, it's a perturbation through vaccination, might we gain some insights into some of these in vivo immune cell interactions that Joe was alluding to between B cells and T cells. And hopefully this data set will provide some interesting insights into that as well. Important to recognize as a backdrop to that, of course, is that the normal vaccine response or infectious response involves a whole bunch of interactions between different immune cells. And although shown here is the classical timeline for developing first the IgM, then the IgG memory antibody response, these are responses that, of course, are much easier to measure and are the typical way by which we gauge vaccine responses. The cellular responses that are taking place here, their interactions, this is just highlighting the busy cell-to-cell type communication that results ultimately in the generation of the memory, humoral and cellular-based responses that confer the important protection down the road. So it's with that kind of view that we were interested in trying to capture, as Joe had shown, both humoral and cell-based responses. This study, which was carried out at Penn, took advantage of a very motivated cohort of individuals, healthcare providers who also have MS and on anti-CD20, who were keen from early on, as they were the first to get exposed to the vaccine, to provide samples, which they were willing to give at 5 different time points shown here prior to the initiation of the vaccine. And again, these are all RNA vaccines, both Pfizer and Moderna. The time point of 10 to 12 days after the first vaccine, then time point 3, the time of the second vaccine dose, and then another 10 to 12 days after the second, and 25 to 30 days after the second. And we collected samples both for serologic type analyses as well as cell-based analyses. These would be the traditional antibody-based responses. We added to that neutralizing antibody assays in collaboration with the Hensley and Bates labs at Penn, and cellular immune responses. We were able to look both at antigen-specific memory B cells using a tetramer-like flow cytometry platform, and then activated T cells, both CD4 and CD8, both by multiparametric flow cytometry, and also the antigen-specific AIM assays, activation-induced molecule assays that Joe had nicely presented. This is the top-line anti-spike antibody data, shown here the trajectories over these 5 time points of healthy controls on the left, and then to the right are the MS anti-CD20 treated. You see here much more heterogeneity, as Joe had shown, and a delayed kinetic, which is emphasized when we look at the right-hand side here. The same data essentially represented now as the spread and the average in the histograms, showing you that there's both a decreased overall magnitude, but also a delayed kinetic of induction of the maximal antibody response in the anti-CD20 treated MS patients. We see very similar results for the anti-RBD antibodies, and the antibody responses, both the spike and IBD, correlated very well with the viral neutralization assay results. The observation of a correlation between B-cell reconstitution and longer anti-CD20 interdose interval and the antibody response is affirmed here. You can see on the left the spike IgG responses, on the right to the RBD IgG antibody responses, in both cases at that last time point 5. And you'll see here plotted on the x-axis are the proportion of B cells at baseline. And as that number increases, you see higher levels of antibody measured. The gray little circles here represent the healthy control population. They're all showing, of course, in association with the normal or high proportion of lymphocytes, B lymphocytes at baseline, high antibody titers, and in orange is the spread of the MS anti-CD20 treated patients. And this percent B cell at baseline, of course, is also correlated with time from the last infusion. In this case, all these patients were treated with ocrelizumab as their anti-CD20 treatment. So reinforcing this time-related issue. When we look at memory B-cell responses, using again this tetramer spike antigen-specific memory B-cell measurement, the healthy control trajectory is shown on the left, the substantial attenuation and delay in the kinetics shown in the middle here for the anti-CD20 treated patients, and now superimposed the healthy controls and the MS at the bottom. So you can see that the magnitude also here is very, very different. Even though you get a little bit of a bump, there's a substantial attenuation when you consider the proportion on the same scale of the generation of these spike antigen specific memory B cells. And the same pattern is true when you're looking at the RBD specific memory B cell responses. What about the T cell world? So the first was an assessment using multiparametric flow cytometry and what's referred to as concatenation of the multiparametric flow cytometry data. So what you're seeing here across the top from time point 1 to time point 5, at the top are the healthy controls. So all the data of all the healthy controls at each time point basically collected together, concatenated, and shown here for the, in contrast to the MS anti-CD20 treated cohort just below. And because we're using multiparametric flow cytometry, we can assess, using this approach, the populations, as we refer to them as clusters or microclusters of different CD4 T cell subsets in this case, and track them over time and compare their kinetics in the patients on anti-CD20 versus the healthy controls. These are several different clusters, in this case metaclusters. I'll focus your attention on metacluster 7, which happens to be the Th1 interferon gamma positive T effector memory CD4 T cells. And what you can see here very nicely, both for the healthy controls and for the anti-CD20 treated patients is the initial bump that they get with the first vaccine. That settles down a bit. They get the second bump with the second injection, which then settles down again. And you can see that there's a little attenuation here of the CD4 T cell response, but for the most part, the patients are mounting quite adequate CD4 activated T cell responses. This is not yet looking at the antigen specific level, but here we're looking at the AIM assays, spike antigen specific CD4 T cells, shown as an example of the dot plots on the left, as Joe had also shown. And here the summary again, showing the trajectories of the healthy controls in gray and somewhat attenuated, but still very nice responses, the first and second bump to the vaccine in the anti-CD20 treated patients. And superimposing the data again, you can see this attenuation, but compared to pretreatment, a very robust CD4 T cell response in the anti-CD20 treated patients as well. And this attenuation in the CD4 T cell response could largely be attributable to a decreased T follicular helper cell response, the TFH cells known to be important in terms of interacting in the germinal center with B cells. Here, what we're suggesting is that B cell presence is important for the normal development of the antigen-specific T follicular helper cells. And that again speaks to not just a unidirectional T cell to B cell help, but a bidirectional interaction between the B cells and the T cells as sort of an insight into the interactions. I'm skipping a similar concatenated depiction of the CD8 T cell subsets. But here you get the summary at the level of the AIM assays again. And what's interesting here, and this was already alluded to in Joe's results, is that in fact, the anti-CD20 treated patients are mounting more substantial, accentuated CD8 T cell responses all the way down to the spike antigen CD8 antigen specific responses. And you can see here now the orange at the subsequent time points, which are the anti-CD20 treated MS patients, are mounting more substantial responses of the CD8 T cells. These are capable CD8 T cells. They express pro-inflammatory cytokine, they express granzyme B and CD108, and would be viewed as those types of cells that are expected to be relevant for the antiviral protection. We, of course, in these numbers cannot show any correlation with protection, but one notes that in the cancer world, people have shown that these types of CD8 positive, granzyme positive, CD107 positive, pro-inflammatory responses are associated with protection and, in fact, survival in the context of SARS-CoV-2 and COVID-19. So what I'm highlighting here, and again, underscored by the CD8 T cell results, is the role, as one can interpret it, of B cells in the in vivo interactions with CD8 T cells. And one interpretation here, as we think in this kind of summary cartoon that has the B cells in the center in this sort of yin and yang depiction of cells that can be more pro-inflammatory and enhance the activation of pro-inflammatory myeloid and T cell responses versus B cells that may have more regulatory properties that may downregulate responses of the respective T cells and myeloid cells. Is it the case that in the context of vaccine responses, certain antigen-specific B cells are relevant to the induction of TFH responses? Is it the case that in the case of the B cell CD8 T cell interaction, there's actually some regulatory role whereby removing the B cells with anti-CD20 allows for a greater CD8 T cell response? This is one of several possible explanations, which we might get to, but I think again, allows us to get some insights into the in vivo cell-cell interactions as this sort of analytic vaccine paradigm. So as a takeaway, as you've heard before, substantially attenuated or even abrogated RNA vaccine antibody responses in these anti-20 patients. Timing matters. There's quite an intact CD4 T cell response down to the spike antigen reactive level with a mildly attenuated TFH response explaining the slight decrease. There is an enhanced spike reactive CD8 T cell response, and together providing some important insights that may help shape guidance with the papers that you, or the work that you've seen from the previous two presentations, and some more that's coming out in the literature to help us with guidance for patients, and maybe also provide us some novel insights into these in vivo cellular interactions. And the easiest way to properly acknowledge this, which was a multi-lab, multi-individual effort is shown here. We collaborated particularly with John Ware's lab, with Socrates, one of the fellows there, and Mihir Kakara, a fellow in my lab, as the co-lead authors, and several individuals here as the senior authors. We did not have funding for this, so as you can see here at the bottom, multiple different entities contribute a little bit, as shown here, for us to be able to do this. And I'll stop here, and if we have time, happy to take any questions. Thanks.
Thank you, Amit. Is there any questions?
Actually, me, so I really enjoyed that. That was really fun, and congratulations. So what are you currently advising in clinical practice at the moment in context of vaccination, or are most of your MS patients vaccinated now and that's irrelevant? Or maybe even in context of the third dose, assuming that's where, sounds like that's where the US is going at the moment. So in short, what are you advising in your routine clinical practice today?
Yeah, so it's sadly not the case that all MS patients are vaccinated. And with anti-CD20, as the initial results were coming out that people may not mount antibody responses, that actually fueled a little bit of the vaccine hesitancy. What's the point in getting it if I'm not going to mount a response? So I think the results that show that there is a T-cell response, and again, short of having confirmed that that's associated with protection, are important for us to be able to indicate to patients that getting the vaccine is important for you. It's not a waste of time, and almost certainly be protected. And the protection we think that is arguably most important is from severe infection or severe COVID complications. And everything we know about CD8 antiviral responses would suggest that these types of responses that we document, and Joe showed as well, ought to be protective from severe infection. The presence of antibodies for this particular virus is important. The neutralization of the virus through the spike protein engagement prevents cells from getting infected in the first place. But once cells are infected, and of course, when you're exposed, the vaccines don't decrease your exposure, some cells will be infected. And if they are, the antibodies are no longer useful. Having a good T-cell response will kill infected cells and prevent further spread. So it's plausible to think that you'll get protection from severe outcomes, but you may not sterilize the virus as quickly. You may be a carrier for a little longer. So practically speaking, we've been encouraging people to get the vaccine and the booster shot. And of course, if possible, prior to going on the anti-CD20. If on anti-CD20, trying to get it now, I think we have clear data to support what we suspected, which is the timing matters relative to the infusion. So to wait, if possible, several months or longer after, and then to wait, if you're already on anti-CD20, for at least 4 weeks prior to getting your next infusion, because you would remove the B cells while they're still playing likely an important role in ensuring the strongest vaccine response that you can get. And at the same time, to consider maintaining protection, especially around non-vaccinated people, given that you may be a carrier for longer.
Okay, thank you, Amit. Thank you, Helen. Any other questions? No? Well, actually, I have one, because, like we're talking about timing, because those CD8 cells that are produced after vaccination are, of course, again, spike. But in the viral infections with SARS-CoV-2, the main antigen presented by class 1 after infection would be nucleocapsid. It's a bit like HIV, when, is not the target of the CD8, but GAG is. So I'm just wondering, is those CD8 anti-spike will be there, but what do you think in terms of the timing of those response, like in the case of a natural infection after vaccination? Because they are not directed against nucleocapsid.
Yeah, so I think that's a very important question. And that's why I'm cautious. And in fact, Penn tries to advertise stuff, and I kept on scratching out wherever they said, we've proven that that protection is conferred, right? The data from the cancer world does suggest that measuring these cells and their presence correlates with protection from serious COVID infection and mortality from COVID. So that's reassuring. But I think until we do the studies to be able to measure the different T-cell directed responses and correlate them, with protection, we won't be able to formally make that case. The challenge, of course, is that what can be done with antibodies on 10,000 individuals to be able to correlate with protection is not practical with the cell-based types of assays that Joe and I described.
Yeah, I think they are protective. Okay, thank you. Is there any other questions in the chat? I don't see anything. No, I can't see anything. Well, we're on the top of the hour. So what iWiMS wanted to do was to thank our speakers, Celine, Michella, Joe, and Amit. We really thank you. It was a great session. And we want to thank you, all the participants as well. Thank you very much.
Thanks, Fabienne. Thanks, everybody. Take care.
Thank you, and you can share the information that this was recorded and will be available on our website.
Yes, that's right. Sorry.